In our intro video, we tried to answer the question, what is anatomy? And that is a relatively difficult question to answer because anatomy has several facets. And we're going to look at each of those facets to a varying degree within this class. So anatomy is identifying all of the different structures and parts of the body and then looking at the relationships to one another. And it's the relationships that are important because that is how we maintain homeostasis, which is essentially how we stay alive. So one way to cut the body up into individual pieces is to look at how it was created or what it's made of. And we usually start at the chemical level for that. Chemicals are then put together in vastly different arrays and ways to create molecules. Those molecules then are attached to one another again in very, very different ways and in different combinations to give you a cell. And this sort of gives us our first anatomical facet. And that would be, what is a cell? And in order to answer that question, we then have to look at the field of cytology. And cytology is where we look at the cell and we further break down the individual components and then ask ourselves the question, well, how does one component of the cell interact with the other components to keep the cell alive? And so you're going to see a picture very similar to this within the different presentations and the slides that you have. And this is the cell, and we're going to pay a lot of attention to the cell in chapter 2, but just very quickly, some of the most studied and well understood organelles. We have the nucleus here, and then there's actually a non-membrane bound organelle inside the nucleus, and that is the nucleolus. And then attached to the nucleus, we both have rough ER over here because attached to it we have ribosomes. And then over here we have smooth ER, and the smooth ER does not have ribosomes. And here we can ask a very important question. Well, what difference does it make if the ER has ribosomes or not? Well, ribosomes are the micro machines within your body that create proteins. So when we see the ribosomes on the rough ER, we know that that rough ER is actually creating proteins that will be incorporated into the membrane or possibly secreted outside of the cell. Smooth ER, it doesn't have the ribosomes, so because that micro machine is missing, then we know that the smooth ER does not create proteins. It's actually involved in the synthesis of different fatty acids that you're going to use as components of the cell. Then we look at some of the larger organelles here. We have the mitochondria, which you may realize produce the ATP, which is the energy currency of the cell. And then over here, this very large green organelle, that is going to be your Golgi apparatus, which sort of acts like a UPS system or those Amazon trucks that you see everywhere. This is going to be packaging and delivering the materials that are created by the cell to either different areas within the cell, or they'll be exported out of the cell and created, and will follow different pathways to adjacent cells or maybe even different areas of the body. So that's cytology, and that's going to be primarily chapter 2. Then we have histology. Histology is going to be chapter 3, and chapter 3 is going to talk about all of the different tissues that are created. So if you remember back to what we were discussing, we have chemicals, then we have molecules, molecules create cells, cells then create tissue. And the body is composed of 4 major types of tissues, and we're going to go over those ad nauseum, and you're going to get very well acquainted with all of them in chapter 3. But just for a quick rundown of the different tissues, interestingly enough, as complex as the human body is, we're only made of 4 different tissues. And this is the first variety of tissues. And these are epithelial tissues. So when we're looking at these tissues, we're primarily looking at areas within the picture where we have one cell that is adjacent to another cell that is adjacent to another cell. And if you look, these cells are really adjacent. They're stacked on top of one another, sort of like those houses that you see in San Francisco in the neighborhoods where there's no space between them. And that is essentially what an epithelial tissue is composed of. You have these cells that are adherent to one another, and there's no material between them. Now, you can have a single layer of cells, like you see in this picture, or you can have multiple layers of cells that are attached to one another that create the epithelium. So again, in chapter 3, we're going to pay a lot of attention to naming epithelial tissues. So for a head start, if I have one cell layer that is a simple epithelium, and this is going to be simple cuboidal because each of the cells looks like a small square, and this is going to be stratified cuboidal. Stratified because there's more than one layer of cells, and the top layer, when I'm looking at the shape that I'm going to assign to the epithelial tissue, I always look at the top layer or apical layer. And these cells up here at the top, they look square. They look about as high as they might be long, and so this is going to be stratified cuboidal, this is going to be simple cuboidal. Over here, once again, we have a single layer of cells, and I know that primarily because I have a single layer of nuclei. And so when I'm looking and all the nuclei are representative of an individual cell, then I know I have one layer because I don't have nuclei stacked on top of nuclei like I do over here. So this again is going to be a simple, but in this case, the cell is rather high. It's a lot taller than it is wide, and so this is going to be simple columnar. Now you might notice that you see some things hanging off the top of the cells, and these, to me, they look like hair-like structures, and so these are going to be cilia. And this is very common in areas where you're moving materials within the lumen, and the cilia are going to be in one direction, and they're going to push the materials inside of the tube or the structure one direction. Now, if I come down here, I see that I have another stratified epithelium. I've got a nucleus, and then it's stacked on top of this one, which is stacked on top of that one, which is stacked on top of that one. So this is definitely a stratified epithelium. However, when I go to assign a shape, notice that this looks sort of square, this almost looks pentagonal. This one doesn't really have a good shape. This one looks like it might be a little columnar with its one to the side, but none of them, or I should say all of them, do not reflect the same shape. They're sort of disorganized. And I'm going to tell you that this is transitional epithelium. Transitional epithelium is a stratified epithelium, but it does not have a uniform shape in the top layer. Now, this makes it unique, and anything that's unique then becomes special. And so this is transitional epithelium. It is special because of the things that I just told you, but in addition to that, it's located in only one area of your body, and that is within the urinary system, and that is going to be in the bladder. And so what this tissue is capable of doing is actually stretching out, and so as the bladder fills, this epithelial tissue stretches out and accommodates the change in the shape of the bladder while yet maintaining the ability to block the reabsorption of the urine, which is something you wouldn't want to do. I mean, I just spent probably large amounts of ATP creating the urine and to ship these toxins out of the body, I don't want them leaking back into the body as I've just caused them to be leaving the body. I want to urinate them out and get rid of those toxins so that I don't have neurological damage. From here, we are going to go to gross anatomy. Now, gross is not like, ew, that's sickening. Gross anatomy simply means large in size. So what we've been looking at has been relatively small. We started out with chemicals into molecules into a simple cell or a single cell. No cell is probably that simple. And then we took those cells and we put them together to create tissues. And then from those tissues, those 4 different types of tissues, which we haven't finished talking about, those tissues will come later. We have the ability to create organs. And then those organs are going to be large enough to where we see. Put those organs together, we get organ systems. When we take those organ systems, then arrange them, we get an organism. And the whole organism is definitely visible. So it's gross, it's large. Now, when we think about gross anatomy, this is something that we don't need instruments to help us see. And so we have things like surface anatomy, where we look at the surface of the body and we can find individual regions, and we can actually see maybe perhaps grooves or clefts. We might be able to push on it and actually feel bones. We might feel the inferior angle of the scapula, and then we might be able to feel the vertebral border of the scapula. So these things are very, very large. We can touch them, we can feel them, we can see them. And so surface anatomy is one of those things where we're going to look at the surface of the body, delineate different regions, and then be able to identify those structures underneath of that. This sort of goes with regional anatomy because we'll be naming each of the regions, gluteal region and the lumbar area. So all of this then will be broken down into different regions so that we can focus our attention on a particular area. For instance, I may not want to try to comprehend the entire abdomen. I may want to break it into regions: epigastric region, umbilical region, hypogastric region, and then focus on that particular region. And you might ask, well, why don't I want to look at the entire abdomen? Well, if the person has pain only in this area, you don't care about this area. If this area is not in pain, then there's nothing wrong, and we probably don't need to focus our attention there. So we're going to focus our attention in the region where that pain is located. And so we can break the body up into different regions and then be concerned only with that area where there might be a problem. Now, systemic anatomy is somewhat associated with this. When we look at a region, we might have the organs to a particular system of the body. And when we look at the abdominal region, most of these organs in here are part of the digestive system. So we have the liver, and we have the stomach, and we have the large intestine, and we have the small intestine. All of those are going to be organs within the digestive system. So systemic anatomy is when you look at a group of organs, or the items that you're looking at are all operating or functioning within the same system. Now, there are times that some organs will actually participate or function in several different systems. So you may have to look at that organ in a couple of different ways to try to figure out, where's the problem? Is the problem with the functioning within this system, or is the problem with functioning in a different system?